So yeah, my name is Upschulte. I'm a PhD student in the research center in Jülich, and I mostly work with Timo and at Denormalization. And today I will talk a bit about how to synthesize training data for cell detection. Also, one side note, this is one of the works that I could start while visiting Montreal. So there I visited specifically Evans and Blake Richards. So thanks again to my supervisors, of course, and to Highball for making that possible. All right, with that, some context. I work with the 1 micron version of the big brain data. So this animation you've seen already a couple of times. And let's have a look at one section in particular. If you zoom in a lot, then you can see individual neurons. And in my PhD, it's pretty much my job to make sure that we are able to detect each individual cell, ideally in the entire brain. And even better, it would be if it would work across several brains, of course. So this is what it looks like. We pretty much get the contours for each individual cell and end up with such a segmentation mask that we can then use for several things. The registration is one example that you've seen now. Okay, but if you think about an entire brain, you might be able to guess why this is difficult, because one brain alone is estimated to have around 86 billion neurons, and making a deep learning model generalize to all the possible variety that the cells can come with, that's quite challenging. And yeah, with just manual annotation alone, it's really difficult to make it work everywhere. Okay, that's the architecture that we use for the cell detection. It's called Contour Proposal Network. We published it earlier this year. For those of you who are familiar with object detection, deep learning, it works kind of similar like bounding box regression models like YOLO, but without anchors, just for those of you who are interested. So what we do is we encode the contours of each cell in vector form. And with that, the network kind of has to learn how to draw the outlines of each individual cell. So in reality, it's a bit quicker than that. So it's actually capable of real-time computation, which is of course important for us because we have a lot of data. Okay, but if you have a network like that, you still need training data as usual in deep learning. And there are several ways to do it. For example, you can work with point annotation. So that gives you very little information about the shape of the cell, of course, but it's really quick. Then there are bounding boxes. Because of the nature of our model, because we were working with contours, we can actually directly using bounding boxes to train the model. So that's a bit faster, but still there's a lot of information about the shape lacking. So contour annotations are the best way to do it, but that's, again, really time intensive. So the real problem here is manual annotation does not scale to the large amount of data that we're dealing with. So there are several things that you can do about it, and there are several deep learning topics existing, for example, self-supervised learning and student-teacher models, and many others. But today we will focus on synthetic data. So what you see here is a sort of baseline approach that we tried. So we really just defined an algorithm that gives us shapes, random shapes pretty much, with texture. And if you create a lot of shapes and put them into one image, it looks something like this. And now you can take the detection network and train it to detect those shapes. And if you do something like this, the network has to learn a lot about geometry because it needs to understand that all the little corners and the entire contour region belongs to one single object. And it finally forms some form of understanding of what sort of patterns really form an object and what patterns don't. Because also as a human, if you look at this, you probably also have an intuition about what's one object and what is another, even though it's just random shapes with texture. So for that, it works really well and it stabilizes training and the model generalizes better. But of course, this data looks nothing like a real brain scan. So we were wondering, can we make synthetic data that actually looks like a real proper brain scan? So that's pretty much the task description. On the left-hand side, you can see the mask images. That's what we provide. So we kind of need to know what shapes to expect. And we would like to map that to a realistic version of a brain scan. So luckily this works. And before we go into the model that does this, I would like to have you to have a look at these two images and try to guess which one is fake and which one is real. And I hope this works out for me. Okay, so hands up for everyone who thinks the right one is fake. And hands up for everyone that thinks left one is fake. Okay, I think it's roughly 50-50 on some people that couldn't decide. So to solve it, the right one is actually real, the left one is generated. And if you had a hard time telling this, then I guess the network did something right. Okay, so that's the model that we are using. It's a generative adversarial network. Again, on the left-hand side, we have the mask image. That's what we provide. The generator would try to map the mask image to a realistic version of a brain scan. And the discriminator, as usual, tries to tell whether its input is generated or actually coming from our scanners. The real key point here is that the generator has to respect what's in the mask image. So if we put a cell into the mask image, it should be there in the generated version. Otherwise, we can't really use it for any training. So that's the real challenge that we had here. And this is how we tackled it. So we used spatially adaptive modulation, or sometimes also called denormalization. So that's a deep learning technique that you can use in the generator. What you do here is you take the mask image, and based on the content of the mask image locally, you will change the intermediate features inside of your generator. So in other words, where you see a cell pixel, where a cell should be, you will modify your intermediate features in one way, and for the background, you will modify your features in another way. And with that strong signal, it's more likely that the generator actually picks up the signal that you provide in the form of the mask image and will condition whether it's going to be cell or background based on the mask image. So it gets more likely that we get the outcome that we actually desire. This is what it can look like in practice. So this is one of those learned feature changes pretty much. So what you see here visualized is the latent space from which you can linearly derive how to scale and shift your intermediate feature distribution. And what you can pretty much tell here is that cell pixels are just really treated differently from background pixels. And with that, you introduce quite a strong signal into the feature space that the generator can learn to pick up. And this is one more incentive that is required to make it work after all. So it's the content regularization. With that, we pretty much just tell the network that cells should be, in our case, a bit darker and background should just be a bit brighter. It's pretty much a form of distance-based loss, but with a cap to it. So our assumption of dark and bright will never be exactly replicated by the generator. Here are some qualitative examples. So there are two data sets from the Broad Institute and some data from the Big Brain. What you can see is that the instance information that we provided in the form of the mask image is mostly respected pretty well. So we can actually use generated data to train detection models. Of course, there is some possibility of errors there, so you have to treat it as a weekly form of weekly supervision. Yeah. Also, very interesting to note, the distribution of cells, whether it's going to be a very densely packed region or very sparsely packed region, that's entirely up to you. So you can really create the data that you need for your specific segmentation situation. And that's quite an interesting situation because you can really focus on the scenarios that your network struggles with. Also, one interesting side note regarding the 41 data set, it shows blood cells, and we don't really work with blood cells, so we didn't really know what shapes to expect. Also, the data set doesn't come with annotations, so we didn't have the shapes. So we just assumed ellipses, And still the network tries to do its best and make the generated image correlate to the mask that we provided, just with some slight shape error to make the image actually look real. And that was quite interesting to see. Also, in the case of the big brain data, there needs to be there's a lot of things going on in the background. So it's not just homogeneous gray or white or whatever, because there are many structures. There can be artifacts, tiers, vessel structures, and many other things. And all of that is actually captured by the generative model, which we are really happy about. Okay, because we also deal with 3D data in the form of Z-stacks, where we pretty much scan the tissue that we have with multiple focus levels. We also try to generate 3D data, and this is what this looks like. So it works quite well, and the instance information, again, is pretty correct, and we can see here on the right-hand side 3D visualization of the 3D shapes that we have. So we can also use that to do 3D segmentation, which is really nice because 3D annotation by hand is even more cumbersome than it is already in 2D. Okay, and that pretty much wraps it up. If you're interested in this work, definitely feel free to get in contact. You might also want to check out our GitHub, which you can find at celldetection.org. And one final slide, because I always find it interesting to look at out of distribution examples. So some shapes that definitely are not cells. I would like to say thank you very much.